drugs on diet?
Short answer, balanced diet. Diet, well, that is answer is simple because I work with diet and neuroinflammation and cognitive flexibility. So diet is the physical activity, enough rest, attention and care, yes. Attention and care, yes.
I think from your question, there are some diets. Like you rightly mentioned, vegetarian. We are talking about children. Definitely, you will be careful of the drugs you give them at the night. So, if you give them maximum sleep, you help. Do you understand? And even recreational activity,
Please clap for Capstone 1. They very well. Thank you for the presentation. Thank you for your responses to questions. Please keep celebrating them until they get to their seats. All right, Capstone 2. The team leader or the team leader and the leader.
Yes, you can. Sit down.
Good morning, everyone. I'm Ojo, representing Group 2 for our capstone presentation on the topic behavioral neuroscience. What is behavioral neuroscience? It's simply a scientific study of how the brain and the neurons, the nervous system, produces behaviors from simple reflexes to complex emotion and cognition. And it's also known as psychobiology, biopsychology, and biological psychology. And there's an interdisciplinary connection that comes up with this study subject, neuroscience, such as there's a crossroad between biology, neuroscience, and psychology. And what are the core questions? Why do we think? Why do you behave the way you behave? Why is it that when you want to sleep by 9 p.m. and the light comes on, you feel alertness till around 12:01? Why is it that when you are hungry, you are hungry? Why is it that when you see food and I tell you that this is your food and you begin to salivate? These are the things you need to begin to think about when we talk around neuroscience. Who works in this field? Biologists, physiologists, anatomists, psychologists, psychologists, clinicians, and all that. And we have historical milestone in this regard. Phineas Gage in 1843-48. Now, the iron rod pierced through his frontal lobe, and this damage, and from there, his personality was distorted. And this was a milestone that shows that the prefrontal cortex actually governs behaviors and emotion and self-regulation. The Broca's, we also learned about Broca's and the Wernicke's area, and this demonstrates that specific brain region actually coordinates specific language production and comprehension as well. We have the patient's HM, whose hippocampus was removed, and from there he was unable to retain long-term. And from there, we got to know that the hippocampus is actually responsible for long-term memory and memory consolidation. Then Ab's rule that says the neuron that fires together wires together, and we have the discovery of the long-term potentiation as well as then we have the modern era where we use optogenetics, the functional magnetic resonance imaging, and all that to begin to investigate further into this context. Modern tool for behavioral neuroscience. We have the functional magnetic resonance imaging, the optogenetics, the electrophysiology, the gene editing, CRISPR gene editing, and as well as calcium imaging and the computational modeling. Animal models in behavioral research. Why do we begin to use animal models in preclinical studies? You see us yesterday, we went there and we make use of our Wister rats. One of the reasons is that they shared neuronal architecture. The fundamental organization of mammalian brain are actually similar. If you see the organization of human brain and that of rodent, they are of course similar. You see the cerebral cortex, the brainstem, the cerebellum, and all that. And this gives us a good clue on the fact that if we use animal, then we are most likely reproducing the same thing. Then the ethical constraint, many experimental essential for understanding disease mechanism and all that, cannot actually evaluate this thing on on humans. Then the use of animals. Then translational values. Animal studies have driven breakthrough in the treatment of depression, post-traumatic stress disorder, addiction, Alzheimer's disease. Mention them, all these psychiatric disorders and neurodegenerative disease and neurodevelopmental diseases. Animals have been used to have a breakthrough in evaluating these diseases. Then the most common species, we make use of mice, rats, as we commonly use, zebrafish. Now, research is now, we have seen people now using Drosophila melanogaster, even amidst us in our midst. Then the genetic control, inbreed strain and transgenic lines allow specific gene to be turned on or off. We can now use transgenic by studying the first generation and all that. When you have your inbred animals, then you can effectively study all that. Then recent advances, we have 3D brain, organoid, humanized mouses model, and all that. Then the 3 validity criteria for the use of animal models, we have the face validity. How that the model of disease reproduce the same similitude like in animal. For instance, there's a reduced movement and anhedonia in animals, which is actually a proof that we see in humans who are depressed, that you lost pleasure in you lose interest in pleasurable events, and then you are sluggish and you don't want to relate. The deprivation of social interaction, where we can easily check in rodent in social interaction model. There check with force swim test and tail suspension test, how that the animal will lose interest in flapping its hand and it increases the time of immobility. Then we have the construct validity, how that the mechanism of action that we see in rodents, in animals, is actually similar to that of human. For instance, the HPA axis that is dysregulated in animal, the corticosterone level in animal that are depressed compared to the cortisol in humans. Then we have the predictive validity, how that the treatment actually produces a similar effect to that of humans. The ethical principles, the replacement, the reduction, and the refinement. Animals, the non-animals alternative, when it's scientifically possible. And reduction uses minimum number of animals. We do ethical approval where they tell you cannot use more than this number of animals and all that. Then refinement, it minimizes pain, humane killing, and all that. Then we have the regulatory oversight, the sex and biological, all that. In 2016, NIH is now suggesting that in grant research that you should use both strains so that you can actually evaluate the real difference in that. The role of neurotransmitters in behavior, my dear colleague will continue with that.
All right. Thank you so much. You cannot go. What are neurotransmitters? Neurotransmitters are chemical what are the key neurotransmitters in behavior?
You can just summarize what you want to say. Because time is it's a win-win for you. Just summarize what you want to say.
Why the balancing both and inhibition. We also have the serotonin, which modulates mood, appetite, and social behavior. We have the dopamine, which drives reward, and motor. We have the norepinephrine, which regulates another attention and the stress the regulation of the motor center. Then we have acetylcholine, which controls learning and rapid activity. Now, what are the neurotransmitter imbalance in psychiatric disorders? I'll talk about it. We have the Parkinson's disease. This is a neurodegenerative disorder or the pathogenesis is from the degeneration of dopamine neurons
Should we skip that?
in the basal ganglia.
Should we? Because of time. Fear, anxiety, and amygdala. We can't skip it.
We also have addiction. Repeated use of drugs.
Yes, amygdala.
Release of dopamine.
Yes, yes.
The messenger dopamine system. There is
Just try and correlate it. That's all.
That's the mental tegmental area to the nucleus accumbens, from the nucleus accumbens to the amygdala, from there to the hypothalamus. So the rotation of this movement or this pathway leads to addiction. Then we also have Alzheimer's disease, which is from the position of amyloid beta like those angles, which leads to the generation of cholinergic neurons in the brain. Further leading to brain atrophy. For brain atrophy, we have symptoms like cognitive impairment and other things like memory loss. So my lecture will continue from that. Thank you.
How? Move forward, move forward, move forward, move forward, move forward.
Thank you very much. I'll go to which is the take-home section. The take-home section, which is the behavioral science linked with behavior through rigorous multiple investigation from Jesus section. And then I want you to know this take-home is a period.
Questions, please. Questions from students. Questions or comments? Questions or comments?
Yes, sir. All right. The biological factors, you mean biological, the mechanism of action, right? All right. In a normal system, in a physiological stress, physiological stress, through negative feedback, there's a relation. The hypothalamus hypopituitary adrenal axis. When the body senses stress, then the hypothalamus begin to secrete adrenocorticotropin releasing hormone. Then that stimulates the pituitary gland, the hypophysis, the adenohypophysis, particularly the anterior pituitary gland, to begin to stimulate the gonadotropin. The hypothalamus initiates gonadotropin releasing hormone. Then it stimulates the anterior pituitary to begin to release gonadotropin. Then this then stimulates the adrenaline to begin to stimulate the release of corticosterone. So this maintains muscle alertness, increases heart rate, and all that. But once that physiological state is regulated, through the negative feedback, there's a signal that's sent back to the hypothalamus that stop producing the gonadotropin releasing hormone. Then the stress comes back to normal. But in disease condition, if you want to replicate that, then you can begin to keep administering corticosteroid into the system. So that system will be dysregulated because the negative feedback will be disrupted. Then the body will begin to maintain stress. And that begin to result to excitotoxicity because the neuron begin to overfire. And that will ultimately result to neurodegeneration.
Second question. She asked a question.
Okay, what's the second one? The neuronal changes. Okay, I just summarized that from my last statement. When this happens, the part of the brain that's responsible for emotion, mood, and all that, they begin to overfire. If you don't, and that's the reason why when you stress yourself, and you are not giving yourself rest, the body begin to dysregulated, and that's when you begin to feel headache, dizziness, and all that, because the neurons are now overfiring. And in a normal physiological settings, when you now give yourself rest or you sleep, or sometimes when you take medication and you relax, then it returns back to normal. So it often results to overfiring of this neuron in order to meet up with the stress condition.
What happens in human and what correlated behavior we expect in animals to take, for instance, a major depressive
Disorder.
and hedonia. So we cannot put it as hedonia in animals.
Anhedonia,
We do sucrose.
preference test.
Yes. So what some of these diseases you have mentioned, what are the symptoms in human and what is the correlated behavior in animals? I think
Okay. We actually mentioned it clearly. Loss of pleasure is one of the symptoms of depression. Now, and one of the ways through which we can replicate that in animals is that we try to know if the animal is still interested in pleasure. And one of those, the way we can check that is by administering, giving them access to sucrose solution and normal water. So in animals that already lose pleasure, that they've depressed, they will take less of sucrose solution compared to, so it shows that they are already losing pleasure in things that they have enjoyed in the past.
Nothing on amygdala. Okay, time off. We've exceeded the time for questions. All right, please. Can you give them the big hand that they are saying? Absolutely.
Emotional regulation. Limbic system. Since you just look percent
so far you have an idea.
Good morning. My name is currently a student in Lautech, Department of Anatomy. No, she's not presenting. I'm the team lead for Group 3.
Good morning, everyone. My name is Beloved Adebayo. Hi. Good morning, everyone. My name is Beloved Adebayo once again, and I'll be presenting on Beyond Movement, the cerebellum's emerging role in cognitive processing and neuropsychiatric disorders. Now, this is a study to challenge what you have known for over 200 years. It's a study to show that the cerebellum is not only concerned with motor coordination, but beyond movement. This is a traditional view. The cerebellum has always been understood to control motor coordination only, and this was supported by the study carried out by Florence in 1820, a French physiologist who isolated the cerebellum and understood or observed that the cerebellum without the animals without the cerebellum didn't have muscular coordination and equilibrium. Also, Babinski in 1899 understood from the patients that he observed that had cerebral lesions that they could not execute complex movements, and this has solidified this knowledge over the 200 years. Now, this is what we are bringing. How can the cerebellum, which contains about 50% of all the brain neurons and only 10% of the total brain volume, only be for motor coordination only? The answer is simple. It is more than that. Functional imaging has shown that there's bidirectional connections to the prefrontal cortex, and it links to the limbic system, which is in charge of memory and also language. It also is connected to the parental areas of the brain. It's concerned with roles in cognition, in emotion, and executive function. The aim of this study is to review the cerebellum's role in cognitive processing and examine its implications for understanding neuropsychiatric disorders with a focus on the functional neuroanatomy. The objectives have been divided into four: describing the functional neuroanatomy of the cerebrocerebellar circuit and its cortical projections, reviewing evidence linking cerebellar structure and activity, examine cerebellar dysfunction in ASD, schizophrenia, and ADHD. And the fourth one is proposing translation insights such as academic review, such as therapy, and also clinical diagnostics. So the first one is talking about the anatomy, the second one is talking about the functional view of the cerebellum, the third one is talking about the pathology, and the fourth objective is talking about the clinical diagnostics and also the translational insights. Moving on to the cerebellar circuit, which is speaking about the anatomy. We cannot speak about the function without talking about the wiring. The cerebral cortex is connected to the thalamus and is connected to the cerebellar cortex also. So from the cerebral cortex, we have the thalamus and we also have the cerebellar cortex. It's called the bidirectional flow. It's also called as the cortico-palato-cortico-thalamal tract. Now, when you think about an object, when you think about the reasoning, the cerebral cortex is in charge of firing signals to the pons, which is a primary relaying function, and the pons fires the signals directly into the cerebellum, and in the cerebellum we have cells such as the Purkinje cells, which are the major cells of output in the cerebellum. The Purkinje cells now sends inhibitory signals into the dental nuclei, which is responsible for the non-motor cortex, and also the ventral part responsible for the motor cortex. The cerebral cortex is responsible for refining, timing, and execution of these signals so that they can be carried out correctly by the cerebral cortex, which is the prefrontal cortex, the parietal cortex, and the limbic cortex. Moving on to the next. Moving on to the next. Moving on to the next, cerebellar contributions to cognition. There are four major cerebellar contributions to the cognition. So now you know that the cerebellum does not only involve in motor cognition, it's involved in working memory, attention, language, and executive function. And it has been proved through studies that when the cerebellum is dysfunctioned, the working memory of the individual that is dysfunctioned won't be able to work well, and the attention that the person has won't be concentrated. And the language in the sense that when a person with a dysfunction in the cerebellum, maybe like cerebral lesion, is affected, the person won't be able to understand semantics well, won't be able to attribute correlation to words, and executive functions, planning, creativity, flexibility, and abstract reasoning also are affected when the cerebellum is not working properly. Thank you.
The second one is structured cortical cerebral thalamic circuit. So the structured cortical cerebral thalamic with smaller cerebral body, especially in the back. Motor, social, and executive impairment is the regular disorder. So here we see the concept of recognizing the so now the cerebellum appears to be the common neurological link between the motor densities and cognitive, social, yeah. So the third one is attention deficit hyperactivity disorder. So the attention deficit hyperactivity disorder, we see that the cerebellum is not only participate in motor coordination with motor attention.
Or it can brush straight,
Motor
instead of
as a tiny organ and has influence in our attention, how what we are attentive to. Okay, so translational relevance and expected outcome. Now that we have spoken about the fact that the cerebellum does not only participate in auto coordination, what is the expected outcome or what does this project now, how does this relevant to us in this model? Conceptual model, mapping cerebellar subregions to specific cognitive function in new functional atlas. So right before now, we have maps, we have the motor maps for the cerebellum, for the core cognitive map. So with this study, we could come up with a cognitive map, where we call early diagnosis. Now we can diagnose this, this cerebral dysfunction area with this study. Then neurostimulation, the transcranial magnetic stimulation or transcranial direct current stimulation targeted on
Thank you so much. Please participate. Question. Question from two participants. Let's see.
Functional magnetic resonance imaging.
Question.
Which part of the cerebellum is impacted when it comes to ASD and ADHD?
Good question. I'm not validating anything, so stop looking at it. Just answer and explain.
Containing the locus behind and behind.
And see your
Sorry, can you come again with the question?
Is it observed in all individuals or just in some? Yes. The last question. I also within 5 minutes. Did you get it?
Thank you so much for that question. Earlier when I was presenting, I mentioned that the cerebellum contributes to not just motor coordination, but also thinking and also coordination. And also refining movement. So how the cerebellum contributes to muscular movement?
Okay, sure.
When it is in dysfunction, for example, the study carried out by Da Vinci in 1892 showed that when the patient with cerebral lesion, when they were observed, they could not carry out complex movements, and it made that observation inextreme. So it means that when the cerebellum, which is in charge of motor coordination, is affected, they won't be able to carry out very complex movement, and also it means that the signal that is supposed to be carried or sent to the muscles won't be available. So it contributes to muscular stiffness in that sense. Thank you.
Thank you for the presentation. Please keep them in the round of applause while they take their seat. Captain 4 of 5. 4, Captain 4. Team leader and the member.
Good afternoon, everyone. Dr. Wala is honestly for
My name is Aki Folaya, I'm a PhD student at Sheraton of Anatomy in Zimbabwe.
My name is from the University of Malaysia. My name is Akiko Jasper from the University of Anatomy Department in Zimbabwe.
Yes, just click this.
Group of neurological disorders that causes progressive loss of neurons in the central nervous system. Okay, thank you. In both the central nervous system and the peripheral nervous system. And basically, they are characterized by collapse of neural networks with neuronal loss leading to breakdown of communication circuits and subsequent impairment of memory, motor, as well as sensory function. Now, there are various types of neurodegenerative disorders, but for the purpose of this discussion, we'll be focusing on just two of them, which are very common. We'll be talking about Alzheimer's disease, which is a chronic progressive degenerative disorder that is characterized by cognitive decline, which is usually driven by the pathological accumulation of neurofibrillary tangles and amyloid beta plaques. In terms of the neuropathology of the disease, you have it is defined by extracellular accumulation of amyloid beta plaques, intracellular neurofibrillary tangles are usually composed of hyperphosphorylated tau protein, then loss of synapses and regional neurodegeneration. And these features are most prominently seen in the entorhinal cortex, hippocampus, as well as the basal forebrain cholinergic nuclei. This means that the basic neuropathology is affecting the cholinergic flow. Now, going to the signs and symptoms of the disease. Of course, this we said they are the commonest form of the disease that we will see from day-to-day clinic, although they are not disease entities that people regularly see in the clinic except in the specialty clinics. And they are usually attended to by specialists in the neurology department of internal medicine everywhere in the world. Now, the signs and symptoms vary depending on the stage by which the patients are presenting to the clinicians. Now, in the early stage, you have episodic short-term memory loss. They keep forgetting information from time to time. Uh, there is difficulty in retaining new information. It's usually very difficult for them to learn new things, and then patients may experience problem solving, judgment, executive functioning, and organization skill. They find it difficult to comport themselves. Now, in the moderate stage, the symptoms range from language disorder, impaired visuospatial skills, there is also involvement of neuropsychiatric symptoms like apathy, social withdrawal, disinhibition, agitation, psychosis, and wandering. Some of these patients are found to, when they move outside their, environment without guide, you find out that some of them get themselves stuck at various places. Even inside the houses, you observe that some of them, when they are eating or doing, maybe somebody wants to move to the toilet, they just get stuck at the point, forgetting that this is what they want to go and do, and they can be for hours, expecting somebody to intervene and actually guide them to what function they want to perform. And in the late stage, difficulties in performing learned motor tasks are usually characteristics of olfactory dysfunctions, that is loss of sense of smell, sleep disturbances, and extrapyramidal motor signs like dystonia, akathisia, and Parkinsonian symptoms. And usually, primitive forces, incontinence, and total dependence on caregivers is the last resort. Now, coming to Parkinson's disease is a chronic progressive neurodegenerative disorder characterized by loss of dopamine neurons in the basal ganglia and primarily in the substantia nigra power factor, like a previous speaker has said. Clinically, it is defined by motor features such as asymmetric bradykinesia. When we say asymmetric bradykinesia, slow movement that is usually not the same from both sides of the body. There is rigidity, resting tremor, and postural instability. Now, talking about the pathophysiology of Parkinson's disease. Now, the hallmark of Parkinson's disease pathology is the progressive aggregation of misfolded alpha synuclein protein that forms intraneuronal inclusions known as Lewis bodies and Lewy neurites. This aggregate triggers selective neurodegeneration through multiple interconnected mechanisms. Men- to mention a few, mitochondrial involvement, impairment, lysosomal dysfunction, altered calcium homeostasis, and chronic neuroinflammation. Now, beyond the central nervous system involvement, there are other systems that is involved. Like I've mentioned in the definition, the peripheral autonomic nerves are also affected. The gut-brain axis is also affected, contributing to development of early non-motor symptoms. The characteristic motor deficit result from loss of dopamine in the nigrostriatal pathway. This leads to disruption in the cortical basal ganglia and the thalamic sulcus. Now, the signs and symptoms we have actually met in the previous